Now that you're all energized and exercised, I want to tell you for the next 40 minutes or so about my journey and the journey of another scientist. Both of us relied on public higher education for our achievements. I grew up in Southern California, where my family was a middle-class family. There were 5 children in my family. There wasn't enough money to send us to a selective private university. Public education was what gave me a leg up and gave me the opportunity to do what I'm going to tell you about this afternoon in the scientific world. So let me tell you first about another example of a brilliant scientist, a Chinese-American scientist by the name of Roger Tsien. So I'll go to my... Roger Tsien grew up in New Jersey. His parents and his grandparents immigrated from mainland China, where the family was a very highly accomplished family of engineers and scientists. So Roger had the benefit of that exposure from a very early age. He developed an interest in chemistry and physics, and in his school years, finally in high school, he advanced to such a level that he became the award winner of the most prestigious science competition in the USA for school children. It was called the Westinghouse Science Talent Search, and here is young Roger in high school with his the work that gave him this prize, which provided him with a scholarship that allowed him to go to Harvard University, where he excelled in chemistry and physics. He went then to Cambridge University to do his PhD in physiology. And then, subsequently, he was recruited all over the US, but fortunately, we at the University of California at Berkeley were able to recruit him to be an assistant professor in my department. In his years, both at Berkeley and subsequently, he revolutionized our ability to look at molecules in living cells. He devised specialized fluorescent probes that allowed us to visualize the flow of calcium ions into cells and into compartments of cells while they were still living and responding to physiologic stimuli. He had an opportunity to move to the University of California, San Diego, where he was given a position as a Howard Hughes investigator. Howard Hughes supported his research for the rest of his life. In the early 1980s, a Japanese scientist at a laboratory in Massachusetts, the Woods Hole Marine Laboratory, discovered a protein in jellyfish that had an intrinsic ability to fluoresce. This protein was purified by Professor Shimamura, and he called it the green fluorescent protein. It wasn't clear what the protein did, but unlike any other protein molecule that had ever been described, this protein had an intrinsic ability to fluoresce. The structure of the protein created an apolar hydrophobic interior into which 3 amino acid tryptophan side chains were projected in an electronic configuration that allowed the molecule to absorb ultraviolet light and to emit in a visible range a green yellowish hued color. This was entirely due to amino acids and not to some other organic fluorescent molecule. So that was a curiosity at the time. But in the years after that, another scientist at the Woods Hole Laboratory was able to isolate the gene from jellyfish, from the jellyfish genome, that encoded this protein. And Roger realized that when the gene was available, that it would be an amazing tool to allow one to tag proteins in living cells and to watch as they move around in the cell, dragging along this green fluorescent protein. So he and another scientist at Columbia University, Martin Chalfie, published important papers in the early 1980s where they showed that if you take the gene for this jellyfish protein and you stitch it next to a gene of interest, a human gene, any gene that you wanted to study, you would create a hybrid protein where your protein of interest was tagged with an intrinsically fluorescent molecule. You could then take this combined gene and introduce it into a cell, a human cell growing in the laboratory, or even into an organism. And then in real time, in living cells, you could watch as the green fluorescent probe followed the molecule that you were studying of interest as it moved around in the cell. This discovery and application changed the complexion of modern cell biology. It has allowed, in the years since these discoveries, the possibility to study living cells and to see how molecules move around. But Roger took this one step further. He realized that in the structure of this protein, in the apolar interior, there were other amino acids that bordered this tryptophan residue that influenced the emission spectrum of the molecule. He further reasoned that it might be possible to produce mutations in different amino acid positions that might change the electronic configuration of those tryptophan residues, thus causing the protein to absorb ultraviolet light, but then possibly to emit it in a different wavelength than the original green fluorescent protein. Now, he did this very systematically, changing the amino acids in the interior of this molecule to create an amazing rainbow of different molecules, each of which is based on the original green fluorescent protein, but with mutations that cause the emission spectrum to change to any one of these individual protein molecules. Now, not only is this an amazing result and colorful and beautiful, but the practical application of this was it allowed an experimentalist to take any number of these different fluorescent molecules and tag different other proteins in the cell, and to be able, with optical filters, to observe many different proteins as they move around the cell simultaneously, observing the emission spectrum with different color filters. This, too, changed the manner in which we conduct science at the cellular level. One simple example is shown here. This is a cell grown in the laboratory that is caught in the act of cell division. These fibers, labeled with one of the GFP variants, whose emission is orange, represent filaments of a cytoskeletal protein called actin. Blue was a different probe to label chromosomes, and green was yet another version, GFP, that labels another element called the microtubule, and this structure is the spindle on which chromosomes align to allow, during cell division, the sister chromosomes to be separated from one another. We now have these tools as a result of Roger's genius, and it has changed the way that we as scientists can investigate proteins of any nature in almost any cell, indeed in whole organisms. It's possible to visualize a developing embryo and to observe many different molecules in all of the cells of the embryo as it grows and matures. Well, for these discoveries, Roger received the Nobel Prize and continued his brilliant pioneering work in San Diego. Regrettably, all too young, he suffered a stroke and died prematurely in his early 60s, a great loss to the scientific community and to the world. But I want to leave you just with one final message about Roger. All of these achievements that I've just described were done at public institutions where the investment, in this case, of the citizens of the state of California made his discoveries possible. Well, I too was a beneficiary of that investment. I grew up in Southern California in an area called Orange County, but it was long before it was as heavily populated as it is now. There was a river running near my home, and it collected water into a still pond that grew some flora and a layer of pond scum on top of this pond. And one day, for reasons that I don't really understand, I took a jar and I went to this pond and I collected some dirty water. And I, for my 11th or 12th birthday, had received a toy microscope. And I took some of this pond water and I put it on a slide. And I couldn't believe what I could see even with this toy. I could see a world of complexity greater than what I see right now of microorganisms swimming and crawling and bumping into each other, eating each other. It was an amazing, amazing display of life at the microscopic level. One evening at dinner, I was expressing my enthusiasm for what I'd seen, and my father was rather skeptical. He couldn't believe that I could see these things in a toy microscope. So I decided at that point that I was going to get a serious microscope, a student professional microscope. So I used to babysit neighborhood children, I used to deliver newspapers, I used to mow lawns, and I collected my savings in an envelope that I put in the closet in my bedroom, with the goal in 1962 to save $100 US to buy a real microscope. Unfortunately, my mother kept borrowing the money to buy groceries, and I could never get the $100 that I needed. So one Saturday morning, I was so angry that when I finished mowing a neighbor's lawn, I rode my bicycle to a local police station, and I told the duty officer that I was running away from home because my mother was stealing my money and I couldn't get my microscope. So they called my father in, and he went into the captain's office, and there were some words behind a closed door, and he came out quite angry, obviously. But I learned a valuable lesson, because that afternoon, the $100 mysteriously appeared, and we rode to a local secondhand store, and I bought this Bausch & Lomb student professional microscope, which became my pride and joy throughout high school. Every year, I joined my fellow students in a science fair at the local school, at the county, and eventually in the state of California, and we mounted science fair projects for others to see, where we had the opportunity to develop our own interests and to follow our own instincts, reading what papers or books we could read to mount a little project of some interest. This alone was what stimulated my interest in science. And when I got to university, of course, the microscope got put away because the university had better microscopes. And when I was at UCLA, where I was an undergraduate, I learned in my freshman chemistry class about the nature of experimental science in the university, a really serious increase in the intensity of the work compared to what I could do in high school. And so I developed an interest in chromosome replication, and I did well, so that I was able, as a graduate student, to study with one of the great biochemists of the 20th century, a man named Arthur Kornberg at Stanford University. But now I'm going to tell you about my journey after that, because in 1976, I had the opportunity to take a faculty position at UC Berkeley, where I have remained ever since, now going on 48 years. I chose for my own independent path to follow something new that hadn't really been understood at a molecular level, using the tools that I'd learned as an undergraduate and then as a graduate student. So let me now take you on a journey into cells at a different level than you've just seen. We're going to look first at a cartoon of what a cell membrane looks like. So this is a representation of a biological membrane. Many of you who have had an introductory biology class will know that all membranes, those that surround the cell or that surround compartments within a cell, consist of a bilayer, two layers of molecules called phospholipids, here depicted in red. The phospholipid consists of a polar hydrophilic head group, these red dots, that face either the outside of the cell or face the internal cytoplasmic surface of the cell. But attached to these head groups are long-chain fatty acids. These are hydrophobic, apolar. They don't do well when faced with an aqueous environment. And so when you have lipids of this structure and you simply mix them in a test tube, in a buffer, they spontaneously form a bilayer where the head groups project to face the aqueous environment and the fatty acids bury into the inside of the bilayer to provide an apolar environment in which they are satisfied. Now, if membranes were only these bilayers of phospholipids, they would be impermeant to charged species that cannot cross such a hydrophobic barrier. So therefore, cells over 3 billion years of life on Earth have evolved protein molecules, these green structures, that allow cells to communicate with the outside in various ways. They are molecules like channels or permeases that allow ions or other small molecules to be captured and concentrated in a cell or to be expelled from a cell. Or they represent receptor proteins in a membrane to which hormones, molecules like insulin, bind to signal some event to the cell to change some aspect of its metabolism. Now, cells, as you'll see in a moment, are very complicated. Most cells that have a nucleus have many membranes, not just the cell surface membrane. Here, for instance, is one of the most dramatic cells in our body. This is called the beta cell of the pancreas. This is the cell, the tissue in the pancreas that is responsible for manufacturing insulin, that is what we use to control the level of glucose in our body. Insulin is made inside the cytoplasm and is captured into little granules that are surrounded each by its own unique bilayer membrane. Insulin is one of thousands of examples of proteins that must be encapsulated and segregated from other cytoplasmic proteins to enable their export out of cells. The human genome codes for approximately 23,000 different protein molecules, each encoded by its own unique gene. All of those proteins are manufactured inside the cell. They take their instructions from the gene and they are assembled by stitching together long chains of amino acids. Proteins, the catalysts of most chemistry in a cell, have 20 different amino acids. And in different combinations, these amino acids create a long polymer that will eventually fold into a compact tertiary structure to catalyze some structural or enzymatic function. All of those molecules are made inside the cell. But some molecules like insulin, indeed thousands of molecules like insulin, have to be exported out of the cell. They are made inside the cell, but then the cell has to have a way of shipping them outside the cell. So cells have evolved in 2 billion years an elaborate machinery that recognizes those protein molecules that have to be shipped outside of the cell to distinguish them from the rest of the molecules that have to stay inside the cell. Much of what we knew by the early 1970s about how this process works was established by a brilliant Romanian scientist who did his work in New York at the Rockefeller University. His name is George Palade, quite a brilliant scholar in many dimensions, a very cultured man that I had the privilege of meeting when I was a graduate student in Arthur Kornberg's lab. He is seated here beside his favorite instrument. This is an electron microscope. Now, electron microscopes were built initially by German engineers who realized that it would be possible to project a beam, an intense beam of electrons onto a surface of a material, and the scattering of the electrons would allow the surface structure of that material to be deduced at atomic resolution. So it became possible to use this instrument to visualize materials at a microscopic level. Of course, this intense beam of electrons would be too great to allow a tissue to be preserved, to be able to visualize cells. And so what Palade did over the course of about a decade in the 1950s and 60s was, with his team, he devised procedures that could be used to chemically fix tissue slices so that the molecules in the tissue were rigidly held together and wouldn't move at all. He then found that such fixed tissues could be subject to dehydration, where the water was replaced by an organic solvent, and then these solvent-infused tissues could be mixed with a plastic resin that would polymerize to create a solid block. This block could then be trimmed and eventually cut to thin ribbons with a very fine diamond knife. And these ribbons then can be placed in a vacuum chamber at the base of this electron beam, and the fixed samples will resist the electrons that penetrate through the section. The clear areas of cells, electrons will just flow right through, but the structures within cells, particles that may be stained with chemical dyes, would bounce the electrons, and so one would see projected below the screen an image of a kind of a shadow of the cell. And the image that I just showed a moment ago is precisely that kind of image. Well, this technique, in its day, revolutionized our ability to look at cells, in this case, of course, dead cells, but to study the internal architecture of all cells. And so this changed, at that time, the way that we could examine what was going on in a cell. One of the things that Palade focused on was how a molecule like insulin becomes encapsulated for its eventual export outside of the cell. An image that you see here of higher magnification than the one I showed a couple of minutes ago is the last step before insulin is ejected out of the cell. The granule that houses insulin, where it had been encapsulated by a process I'll describe later, eventually migrates up to the perimeter of the cell, and the membrane of the granule touches the cytoplasmic surface of the plasma membrane, and at a key moment that is triggered and regulated, these membranes merge by a process called membrane fusion. This results in the ejection of the internal content of the granule to the outside of the cell without the rupture of the cell at all. This crystalline form of insulin then dissolves, courses through your body, collects in various tissues, where it binds to a receptor on a muscle or a fat cell, and it tells that cell to swallow up the glucose that you've just produced by digestion. That's how insulin works. This event, membrane fusion, resulting in the secretion of insulin, is repeated billions and billions of times in almost all of the cells in your body. Here's a very different example, but uses the same logic. In your brain, or at the junction between nerve cells and muscle cells, small granules, in this case they're called synaptic vesicles, which carry not insulin, but rather chemical neurotransmitters, are produced and gather at the nerve terminal. Some of these synaptic vesicles touch and dock and remain docked on the cytoplasmic surface of the nerve terminal. And when a nerve cell is activated by a change in the potential, membrane potential across the plasma membrane, that membrane potential surrounds the cell, travels down to the nerve terminal, and at a key moment at the nerve terminal, channels that allow calcium ion to flow into the cell, touch these docked vesicles, and cause them, within a microsecond, to fuse very rapidly. Membrane fusion, in this case, very tightly, rapidly controlled. That results in the release, not of insulin, but of chemical neurotransmitters. These are the chemicals that mediate the flow of information in your brain. They are the molecules like dopamine or serotonin or acetylcholine, major chemicals that mediate traffic of information in the brain. In this case, these chemicals, acetylcholine in particular, diffuse across a gap, the synapse between the nerve terminal and a muscle cell, and acetylcholine activates a channel in the muscle cell that eventually causes the muscle cell to contract. Every time you move, this is being coordinated by millions and millions of cells. And in your brain, this is going on at a pace that it's hard to comprehend. Your brain has tens of billions of nerve cells. Each nerve cell can make hundreds or up to 1 million different connections with other nerve cells. This massive network of nerve interactions involves a communication that is absolutely dependent on the controlled fusion of synaptic vesicles to result in the release locally of chemical neurotransmitters. That flow of information, that critical membrane fusion step, is what controls every thought process you have, every memory you have, every emotion you have. All of that is tightly and still largely not understood, but controlled by that membrane fusion event. So bear that in mind as I then return to the discoveries of Palade that allowed us to see how these molecules progress from deep inside the cell to the cell perimeter. In 1974, George Palade won the Nobel Prize, and just after his receipt of the prize in Stockholm in December of that year, he traveled to San Diego, where he gave a lecture at the annual meeting of the American Society for Cell Biology. I had just left my graduate work to go to a laboratory where I was learning about membranes at UC San Diego, and I had the pleasure of watching Dr. Palade deliver a version of his Nobel lecture to a very appreciative audience of cell biologists. Here is what he discovered. Palade, studying the pancreas, was able to show that molecules like insulin are synthesized by ribosomes. Ribosomes are the little kind of sewing machines that stitch amino acids next to one another. Those ribosomes in a pancreatic cell are mostly lodged on the cytoplasmic surface of an internal network of membranes called the endoplasmic reticulum, or ER. He reasoned that these ribosomes must make polypeptides that thread across a bilayer of the ER to fold and form their final structure within the interior space, this salmon-colored interior space, which is called the lumen of the ER. He was able, using techniques that I don't have time to describe, to observe, largely in dead cells, that some of these proteins that are assembled into this canal-like network are captured into small carriers, other kinds of vesicles, that convey their cargo molecules through another organelle called the Golgi apparatus. The Golgi apparatus had been visualized by an Italian scientist by the name of Camillo Golgi in the late 19th century. It was seen in all cells, but it wasn't clear what it was doing. What Palade was able to conclude was this was a kind of a station along the way of a path that led from the endoplasmic reticulum eventually to the cell surface. At the Golgi apparatus, granules, including those that house insulin, are formed by a pinching process, and the granules are then directed to the cell surface where they finally discharge their content by this process of membrane fusion. So this was a beautiful demonstration of an elaborate pathway that had evolved over 2 billion years of evolution. And yet, in 1974, when Palade described his crowning achievement. We did not know anything about how this process worked. We knew the stages in the process, but we knew not a single molecule, not a single protein or lipid that had some role, some functional role. We didn't know the nature of the channel through which these polypeptides must pass. We didn't know how molecules were selectively captured and into vesicles that pinch from one another stage in the pathway. And we did not know what catalyzed this process of fusion, so vital in the pancreatic beta cell and indeed in almost all the cells in your body. So this was a challenge that I decided to take for myself when I had the opportunity to begin my laboratory at the University of California at Berkeley. Now at that time, when I started, 1976, the cell biologists who were the pioneers of Pilates' generation were studying animals or animal cells or human cells grown in the laboratory. That seemed like the obvious thing to study to try to understand this process. But in 1976, the tools to study animals or animal cells grown in culture were very primitive. It would be difficult to use the tools of molecular biology by isolating mutations and isolating genes in animal cells at that time. In contrast, an emerging discipline in cell biology and molecular biology was the use of a very simple single-celled organism called baker's yeast. You all know about baker's yeast, the practical applications of baker's yeast, but you may not know that as a tool to study cell biology, it had unlimited potential to reveal the nature of the mechanisms of essential processes. These are simple cells that can be grown in large quantities in the laboratory. They have a very simple growth style. At the beginning of a cell division cycle, under the right conditions of nutrition, a yeast cell starts to make a bud. The bud grows in preference to the mother cell for the next 90 minutes or so until the bud is the same size as the mother cell, in which case the cell divides by a budding mechanism, and this can be repeated over and over again to produce billions of identical copies of the cells. Now, it has the virtue of a simple growth medium and absolutely identical cells, but most importantly, it's possible to use a simple form of traditional genetics to discover the genes that are required for all the processes that make a cell grow and divide. So we reasoned that it would be possible to use genetics and then biochemistry, because you could produce so many cells, to understand this Pilates pathway. So the way to do this was to expose yeast cells to a chemical that causes mutations, and then to ask for those cells that are defective in the process of secretion. But let me show you first what a yeast cell looks like in contrast to a pancreatic beta cell. So here's a yeast cell, a thin slice, examined in the electron microscope. It doesn't look nearly as exciting as a pancreatic cell, but all the machinery is there. Most of it is a densely packed collection of ribosomes. The background, granular staining in the cytoplasm of a yeast cell are all the thousands of ribosomes that are producing the 6,000 or so different protein molecules that a yeast cell needs to grow and divide. On close inspection, of course, you can see that there are membranes. There's a structure that is the equivalent of the lysosome in human cells. It's called the vacuole. There are membranes that can be seen here that touch the plasma membrane. In this case, these are the ER membranes of yeast. In other slices through other dimensions of the cell, you can see a nucleus with two membranes, just really very much like a mammalian cell. But our attention, and I'll show you the student responsible for this work, was drawn to a collection of small vesicles. These look very much like the synaptic vesicles that I showed you a moment ago, but of course, yeast cells are not nerve cells. They are not secreting insulin. They are not secreting chemical neurotransmitters, but they are producing and secreting the molecules that a yeast cell needs to grow, on the surface of a grape, for instance. There are enzymes that a yeast cell secretes that are captured into these vesicles which are discharged by this membrane fusion process at the surface of the bud. Now, Peter Novick, one of my first graduate students, shown here, busy pipetting away, Peter Novick and I imagined that these vesicles, shown in this slice, may be responsible not only for secretion, but also would be building blocks for the growth of the plasma membrane. Thus, when a vesicle touches and then merges by fusion at the cell perimeter, the membrane of the vesicle would become inserted into the membrane of the plasma membrane, so it would grow as each fusion event occurred. So that simple hypothesis led to a powerful prediction. The prediction is that if you cripple a gene required for the manufacture, for the traffic, or the fusion of these vesicles, that crippling event by mutation would kill the cell. And it's not much good working with dead cells, so you need to have a way of capturing such a lethal event under conditions where the cell can still survive. Fortunately, microbiologists had already figured this out long ago. It's very simple. You can do this with yeast. You can do this with bacteria. What you do is you take a culture, in this case of yeast, you expose it to a chemical that causes mutations, and the mutations spread out among the genome randomly. Occasionally, a mutation will change an amino acid in a protein molecule that will cause that protein molecule to be unstable. Sometimes that instability is reflected in a thermal instability, where the protein works normally, let's say at room temperature, but when the cell is warmed up to human body temperature, that mutation may cause the protein to unfold, to lose its function. There are many such mutations that can be introduced randomly and used to identify essential genes. These essential genes, if mutated in this way, will allow a cell to grow on a petri plate and form a colony at room temperature. But if warmed to 37 degrees centigrade, those cells in that colony will no longer grow. This is really a trivial way of identifying the essential genes in the yeast genome. I told you that yeast cells manufacture about 6,000 different proteins. Among them, 1,500 are known to be essential. That is, if you delete any one of those genes, the cell will simply die. But if you mutate them selectively, you may be able to produce forms of the essential protein that are thermally unstable. So that's what we did. We took a collection of randomly assorted temperature-sensitive yeast colonies, and Peter, as shown here, pipetting away, looked among them for mutants that cause enzymes that normally get secreted outside of the cell to build up inside the cell. That would distinguish such mutations from other mutations that cripple chromosome replication or gene expression. It was a unique phenotype that we could score. Well, the first such mutation we found was near the end of near the beginning of 1978. And at that time, Novick, having found this mutant, we had the opportunity to have Dr. George Pilati visit Berkeley for an honorific lecture, and I was pleased to be able to tell him about our work, but more importantly, the students sponsored a dinner for Dr. Pilati that evening, and Peter told him about his preliminary results with the first such mutant, which we called SEC1, short for secretion 1. Pilati said, well, of course, now you must examine these cells by electron microscopy. And of course, we were going to do that. We raced ahead and did this. And then one day, in the middle of the afternoon, Peter called me in my office. He was down in the basement in the electron microscope room. Very excitedly, he said, come down and have a look at the SEC1 mutant. Now, in contrast to the cell that you see here, SEC1, when these cells are incubated at 37 degrees for a couple of hours, but before they die, they start to accumulate thousands of these vesicles, no longer restricted to the bud portion of the dividing cell. They fill up the entire cytoplasmic volume because the SEC1 protein, being defective in these mutants, prevents the vesicle from docking and fusing at the cell perimeter. So the vesicles continue to be made until the cell basically chokes itself to death. This was very exciting to us, and of course, over the next year and a half, we then repeated this procedure over and over and over again and collected hundreds of such mutations, and in time, we were able to chart a path that's shown here of genes such as SEC1, but now many more genes indicating many more proteins required at each of the several stages in this pathway. And over now decades later, 100 or more such genes are known in yeast, including the genes, for instance, that encode the channel in the ER membrane that initiates this process. Genes that control a bidirectional flow of vesicles back and forth between the ER and Golgi. Genes that control sorting of proteins to the lysosome-like structure, and so on. After we had all these mutants, it became possible to clone the genes and to sequence the genes and to discover what the predicted protein sequences were. But at the time, the genome databases had few entries, and there was nothing in the database that looked anything like the genes that we found. In time, the human genome was sequenced, and not surprisingly, all the yeast genes that we found are found in the human genome. Indeed, the SEC1 gene that starts this story is found expressed in your brain, and it controls the fusion of synaptic vesicles at the presynaptic membrane, virtually the same protein. And this emphasizes, again, the unity of biology. There is an evolutionary conservation. When nature evolves a solution to a complex problem of how to capture proteins for export from a cell, that machinery gets used over and over and over again. And I use this illustration of a cluster of yeast cells, which seems quite different than Charles Darwin, but the fundamental mechanisms that operate in the body of Charles Darwin and all of us are much the same, and they were evolved 2 billion years ago. Now, just in closing, at the time that we were making these discoveries, the biotechnology industry was growing up in the San Francisco Bay Area, and I had the opportunity to collaborate with a local company to help them exploit this discovery for the manufacture of clinically important proteins that would be difficult to isolate by other means. And so, for example, the gene for human insulin, which had been cloned, was introduced into a yeast cell, and now, many years later, 1/3 of the world's supply of human recombinant insulin that's used to treat diabetics is made in giant fermentation plants with growing yeast cultures. Similarly, this company took the gene for the virus, hepatitis B virus, surface antigen, and when that gene is put into a yeast cell, that antigen, which is a membrane protein, collects in little vesicles, and for any of you who've been vaccinated against hepatitis B, you've received little vesicles isolated from yeast cells. Well, let me bring this story full circle. Of course, you recall that microscope that started my journey in cells got put away. Fortunately, my parents saved that microscope. They sent it up to me, to my family near Berkeley. Regrettably, neither of my children ever showed any interest in science, so the microscope sat and collected dust over many years. And on the days after I learned of my Nobel Prize, I and my fellow laureates received an invitation from the Nobel Museum in Stockholm to send in some artifact that reflects a part of one's discovery. In my case, it didn't seem interesting to send in Petri plates, so I dusted off and sent in my microscope. And if you're ever in Stockholm, you can come by and see that microscope. That was what I used to begin my career, including in English and in Swedish, a caption of how I had to run away from home to begin my career in science. Thank you for your attention.